Hello and welcome to the Edupedia world. In the second session of the lecture on sexual reproduction in flowering plants, today we shall discuss about the pre-fertilization structures and events. In our previous lecture, we have thoroughly discussed about the structure of a flower, its various parts, the male and the female reproductive parts, the accessory parts of a flower like calyx and corolla, and also we have discussed about the variations in the structures. So now let's have a quick recap and brush up our previous lecture and quickly move on to the next unit. So, in this figure, you can see the structure of a complete flower, which is also called a perfect flower, a bisexual or a hermaphrodite flower. Such a flower includes all the four whorls, that is, the calyx, corolla, androecium and gynoecium. As you have studied earlier, androecium is the male reproductive part and includes stamens. Stamens are composed of anther, filament and connective. I shall not go into the details here, shall just quickly recollect the terms and proceed on to the next unit. In this figure, you can also see the gynoecium, that is the female reproductive part. It includes carpel or pistil. The carpel is made up of three parts, the stigma, style and ovary. Ovary contains the ovules. Besides the primary parts, there are accessory parts as well, which provides protection to flowers in the bud stage or help in pollination. These parts primarily include sepals and petals. Now let's move on to the topic of our next unit, the pre-fertilization structures and events. First of all, let's discuss about the different sexual stages in the life cycle of an angiosperm. The life cycle of angiosperms follows the alternation of generations. Now, what does this term alternation of generations mean? Here, in angiosperms, the haploid gametophyte alternates with the diploid sporophyte during the sexual reproduction. The first structure in the haploid stage is the microspores. The microspores develop into the male gametophyte and are much smaller than the female spore. Pollen is the male gametophyte and develops in the anther. The other potential start of the haploid stage is the megaspore, which develops into the female gametophyte or the embryo sac. Once the gametophytes are fully developed, they produce male and female gametes. The male gamete is the sperm cell and the female gamete is the egg cell. The mature male gametophyte, that is the pollen grain, develops the sperm, while the mature female gametophyte develops the egg cell. Now let's discuss about the male reproductive pre-fertilization structures and events. So here first comes the anther and the microsporangium. We have previously discussed about the anthers, its structure and function. Let's just quickly recollect what we have learned previously. As you have studied earlier, anther is usually a bilobed structure. Each lobe has two theca. Therefore, a total of 4 theca, also called tetrathecus. Each thecus has microsporangium located at the corners. The microsporangia later develop into pollen sacs that contain pollen grains. In this figure, you can see the transverse section of a typical microsporangium. The wall of the anther is surrounded by 4 layers: epidermis, endothecium, middle layers and tapetum. Now let's discuss each layer in detail one by one. First comes the outermost layer, the epidermis. It is composed of single layer of flattened cells. Inner to the epidermis lies the endothecium. It consists of a single layered, radially elongated cells. It has a thin layered area called stromium which helps in dehiscence of anthers. Next, inner to the endothecium comes 2 to 3 layers of flattened cells called the middle layer. The innermost layer is the tapetum. It is a single layer of cells with dense cytoplasm and prominent nuclei. Now, let's discuss the function of the tapetum. Tapetum provides nourishment to the developing microspore mother cell and the microspores. It also helps in the formation of exine, the pollen wall. It also secretes callase enzymes that dissolves the callose walls of the surrounding microspore tetrads and thus helps in their separation into distinct pollen grains. Many specific proteins of the pollen wall are also secreted by tapetum. Interestingly, these proteins are responsible for pollen allergy. Tapetum also produces an oily coating over some pollen grains called pollen kits. This helps in attracting insects for pollination. Now, after discussing the microsporangium in detail, let's move on to the sequential structures and events that ultimately direct the development of male gametophyte. As I have mentioned earlier, the microspores or the pollen grains represent the first stage of the male gametophyte. Keep this in mind. Now, you must be wondering, how do these microspores are formed? What is the ploidy of the microspores formed? Or what is the fate of the microspores formed? Or what ultimately leads to the development of the male gametophyte? So many questions in mind. Need not worry. We shall answer and explain all these fine details one by one. So let's first begin with the microspores. As the anther develops, the cells of the sporogenous tissue undergo meiosis to form microspore tetrads. These microspores are basically haploid unicellular structures that develop from diploid microsporocytes. The microsporocytes are also called pollen mother cell or microspore mother cell. Microspores are the first cells of the male gametophyte. Each diploid microspore mother cell gives rise to 4 haploid unicellular microspores. The male gametophyte or the pollen grains develop from microspores within the microsporangia or pollen sacs of anthers. The sequential steps in the formation of microspores by a process called Microsporogenesis is as follows. Here you can see the sequential steps of microsporogenesis. In the pollen sacs, there are microspore mother cells which are diploid. The microspore mother cell divides mitotically. In the first stage of meiotic division, the microspore mother cell divides to form 2 haploid cells. In the second meiotic division, it gives next 2 haploid cells. Now, there are a total of 4 haploid cells. These haploid cells are the microspores, which are arranged in a tetrad. Pollen grains are shed at this point. Now, after the formation of microspores, let's move on to the next step of development of male gametophyte, that involves formation of gametes by a process called microgametogenesis. The male gametophyte produces the male gamete or the sperm cell, which fuses with the egg cell. Here we shall discuss the sequential events of microgametogenesis, which leads to the progressive development of the unicellular microspores into mature microgametophytes containing the male gametes. This phase begins with the expansion of the microspore, which is commonly associated with the formation of a single large central vacuole. This vacuolation is accompanied by the displacement of the microspore nucleus to an eccentric position against the microspore wall. In this position, the nucleus undergoes first pollen mitosis, resulting into the formation of two unequal cells, a large vegetative cell, also called the tube cell, and a small generative cell, each containing a haploid nucleus. The generative cell subsequently detaches from the pollen grain wall and is engulfed by the vegetative cell, forming a unique cell within a cell structure. The engulfed generative cell divides once more by mitosis to form two non-motile, remember, non-motile male gametes or sperm cells, which are completely enclosed within the vegetative cell cytoplasm either before pollen is shed, tricellular pollen, or within the pollen tube as bicellular pollen. The lower panel shows the darky stained images of the nucleus. After the detailed explanation of the sequential events leading to the formation of vegetative and generative cell, let's take a brief look at the vegetative and generative cell formation. Here, figure A shows the microspore. Figure B shows the post-mitotic pollen grain with a vegetative cell and a newly formed generative cell. Figure C shows the presence of a single large central vacuole and generative cell pressed against the pollen grain wall. In the figure D, you can see that the pollen grain and generative cell have enlarged. In the next figure, you can see the generative cell is undergoing mitosis. Figure F shows the binucleated generative cell pressed against the pollen grain wall. This represents the two-celled male gametophyte. Now here in figure G, you can see two sperm cells formed by the generative cell. The two sperm cells are still attached to each other but free from the pollen wall. Pollen is engorging but central vacuole is still present. In the figure on the right side of this panel, you can see a mature pollen grain with separated sperm cells embedded in vegetative cell. This represents a three-celled male gametophyte. Now, after the formation of a two-celled and three-celled male gametophyte, let's discuss about the pollen tube formation. So, what is a pollen tube and how is it formed? In most species, pollen grains are released from the anther in a two-celled stage. However, in some plants, pollens are shed from the anther and deposited onto the stigma in a three-celled stage. On the stigma, pollen grains germinate to form a narrow tube called the pollen tube. This pollen tube grows down through the style. The male gametes and the vegetative nucleus along with the cytoplasm is transferred to the pollen tube and is carried at its growing tip. A plug of callose tissue prevents its backflow. So, this was the formation of pollen tube. But now the point to keep in mind is this, that in plants where pollens are released in a two-celled stage, formation of male gametes occurs in the pollens when they are on the stigma, while in some plants the male gametes are formed in the pollen tube just before fertilization. Now, you must be wondering what is the fate of the pollen tube. So, here lies the answer. The pollen tube finally enters the ovule, bringing about fertilization, that is, the fusion of male and female gametes to form an endosperm nucleus. So, what happens to the vegetative nucleus then? The vegetative nucleus degenerates along with the pollen tube after fertilization. So, till now, we have discussed the different structures that aid in the formation of the male gametophyte and the sequence of events that lead to the formation of male gametophyte. At maturity, pollen grains are either two-celled or three-celled. Here in this figure, we describe a two-celled pollen grains showing a larger vegetative cell and a smaller generative cell. The vegetative cell is also called a tube cell. The generative cell or the sperms lie in the cytoplasm of the vegetative cell, surrounded by their own cell membranes and the membrane of the vegetative cell, while the three-celled pollen grains have a vegetative cell and two sperm cells arranged together as male germ unit. Now, let's discuss about the structure of a pollen grain wall. A mature pollen grain wall is a two-layered structure. Exine is the outer hard layer of the pollen wall. It is made up of a highly resistant organic material called sporopollen. Sporopollen is remarkably known for its great strength, elasticity and chemical durability. It can even withstand high temperatures and strong acids and alkaline conditions. So far, no enzyme is known that can degrade sporopollen. You must be wondering, what is the function of the exine? Exine provides protection to gametophytes in harsh environments and provides a barrier against various physical and chemical factors and biological pathogens. It also helps with pollen dispersal and cross-pollination in some species. Exine also enables stigma cells to capture the most appropriate pollen grains by providing species-specific factors for pollen stigma recognition and adhesion. Most pollen walls contain characteristic apertures or pores, commonly called the germ pore. This germ pore is thought to allow pollen tube emergence and changes in grain volume. Next comes the inner wall of the pollen grain, the intine. Intine is a thin and continuous layer made up of cellulose and pectin. The cytoplasm of the pollen grain is surrounded by a plasma membrane. So with this, we have completed the structure and development of male gametophyte and shall now move on to the female gametophyte structure and its development. Before beginning with the development of the female gametophyte, we shall first recall the female reproductive part of a flower. In the previous unit of this session, we have studied about the structure and function of the gynoecium. Carpel is the basic unit of gynoecium. And in this figure, you can see three parts in a pistil, namely the stigma, next an elongated slender stalk-like structure called style, and a basal bulged portion called the ovary. I hope you remember the roles played by each of them. Stigma serves as the landing platform for the pollen grains. Style connects stigma to the ovary. The style also becomes the pathway for pollen tubes to grow from the pollen grains to the ovules containing the female gamete. An ovary contains one or more ovules. Now let's begin with the development of female gametophyte. But prior to this, let's first discuss the basic structure of the ovule or megasporangium. The female gametophyte is called embryo sac and it develops within the ovule. So now you must have got an idea why we shall discuss ovule first. Now the very first question is, where is ovule located in a flower? So inside the ovary, one or more ovules are present attached to a flattened cushion-like structure called the placenta by means of a stalk called funicle. The ovule body fuses with the funicle in a region called hilum, or in other words, you can also say that hilum represents the junction between ovule and funicle. Each ovule is surrounded by one or more protective envelopes called integuments. You can see the outer and inner integuments. Integuments encircle the ovule except at the tip where a small opening called the micropyle is organized. Opposite the micropyle is the chalaza, representing the basal part of the ovule. Enclosed within the integuments is a mass of cells called the nucellus. Cells of the nucellus have abundant food reserve. Located in the nucellus is the embryo sac or female gametophyte. After the structure of an ovule, let's start with the development of the female gametophyte. Remember, the female gametophyte or the embryo sac develops within the ovule. The embryo sac contains the female gamete or the egg cell that fuses with the sperm cell in the process of fertilization. The megasporangium is the first stage of the female gametophyte development. The megasporangium undergoes meiosis to form megaspore in a process called Megasporogenesis. The resulting megaspore in turn divides mitotically 3 times, remember 3 times, to form the female gametophyte or embryo sac in a process called Megagametogenesis. In this figure, you can see the detailed schematic representation of megaspore formation from megaspore mother cell. This process is called Megasporogenesis. Genesis word means formation. First, you can see a megaspore mother cell. This megaspore mother cell is also called megasporocyte. This megasporocyte is diploid and represents the first stage of the male gametophyte. Remember, this megaspore mother cell undergoes first meiotic division to form two haploid cells. In the second meiotic division, it produces next two haploid cells. Now, there are total of 4 haploid cells. These resulting 4 haploid cells are called megaspores. Out of 4 haploid megaspores, only 1 survives and the rest 3 degenerate. Remember. Now, you must be wondering, what is the fate of this surviving megaspore? So, this surviving functional megaspore subsequently divides mitotically first to form a 2-nucleated megagametophyte stage. These resulting 2 nuclei then undergo second mitotic division to form a 4-nucleate megagametophyte stage. Then after, this 4-nucleate stage undergoes third mitotic division to form an 8-nucleate megagametophyte stage. These resulting 8 nuclei position themselves in an arranged manner as shown in the last diagram of the figure. The 2 center nuclei fuse together and are called polar nuclei. 3 cells are grouped together at the micropylar end and constitute the egg apparatus. The egg apparatus in turn consists of 2 synergids and 1 egg cell. The egg cell is the female gamete. Remember, this process of formation of female gamete is called megagametogenesis. These synergids have a characteristic cellular thickening at the micropylar tip called filiform apparatus, which play an important role in guiding the pollen tubes into the synergids. While the remaining 3 cells are arranged at the chelazal end and are called the antipodals. Thus, in this way, a 7-celled, 8-nucleated structure is formed. This structure is called the embryo sac or the female gametophyte. In this figure, you can see the entire process of development of female gametophyte from Megasporangium. Now, I guess that this concept of female gametophyte is clear by now. So, in a nutshell, you have come to know that the female gametophyte is the embryo sac that develops within the ovule, and the ovule is basically comprised of embryo sac surrounded by integuments. So with this, we come to an end of the second unit of this session. Till now, we have discussed the 2 steps of the sexual reproduction in flowering plants, that is the Microsporogenesis and the Megasporogenesis. We have discussed the different structures and events that lead to the development of the male and female gametophyte and the resulting male and female gametes for subsequent fertilization. We have also studied the different locations and role of the structures involved in the development of the male and the female gametophyte. we have learned till now various terms related to these pre-fertilization structures and events. These terms are quite important and interesting. Do try remembering them hard. The figures and the various structures or the sequential events are equally important. Practice them too. So in this slide, you can see the overall comparative picture of the various structures and events that lead to the development of the male and female gametophytes. Just take a quick look at the overall picture and try recall whatever you have learned today. It shall be easier for you now. In our next unit of this session, we shall discuss about pollination and double fertilization. Thank you for watching this video. Feel free to catch up with me with your queries, if any, regarding these sessions at studybiology123@yahoo.in. Hope to see you again.